Hi everyone, this is Miss Romani, and in this lesson, I will take you on a tour of the cell. Place a drop of pond water under a microscope, and you'll discover hundreds of single-celled organisms called protists swimming around. It's incredible. Just one tiny drop of water can contain all these living organisms. They are busy swimming, photosynthesizing, and eating each other. And each of these organisms consists of just a single cell. And that is because cells are the basic units of life. And a single cell can be a complete organism and perform all the functions that keep an organism alive. All living organisms are made up of cells. And so far, most of you have learned about cells that look like this representation of an animal cell, which is a great model to help us understand the structures within cells, and one that we will definitely work with. However, it is important to understand that not all cells look like this. The protists that I just showed you do not look like this, and even most animal cells don't look at all like this. A human body, for example, has different types of cells, like muscle cells, and brain cells, and fat cells, nerve cells, blood cells, each with a unique structure that determines their function. There are actually over 200 different types of cells in a human body, and that's just looking at a single species. The variety of cells is incredible when we take into account different types of organisms that exist. For example, bacteria cells. Bacteria are made up of a single cell, but a specific type of cell called a prokaryotic cell. The main characteristic of prokaryotic cells is that they don't have a nucleus or any membrane-bound organelles inside the cell. They have DNA, but it is organized into chromosomes that are free-floating and not encased in a nucleus. Every other cell, like animal and plant cells, for example, are called eukaryotic cells. Eukaryotic cells have a nucleus, as well as a variety of membrane-bound organelles and other cell structures, which is actually what will be the focus of today's lesson. And once again, let's focus on how structure determines function. So a brain is made up of nervous tissue, which is made up of nerve cells, which in turn is made up of specific structures and organelles that are necessary for their very specific function. So let's take a tour of the cell. The nucleus holds and protects the cell's DNA. DNA does not leave the nucleus, but the genetic information coded in DNA is needed outside the nucleus in order to make proteins. So the nucleus is where DNA is read and copied or transcribed into a molecule called RNA. And RNA can then leave the nucleus through nuclear pores on the nuclear envelope. On the inside of the nucleus is also a dense structure called the nucleolus. The nucleolus is where the cell structures called ribosomes are made, and ribosomes are made from a combination of proteins and RNA. In general, eukaryotic cells are defined by the presence of the nucleus. However, there are exceptions, like the mature red blood cells that are found in mammals. Because they don't have any nuclei, mature red blood cells do not contain DNA and cannot make any RNA. Consequently, they cannot divide or make any proteins on their own, which is fine for red blood cells because they are made in the bone marrow of stem cells, and those stem cells do contain a nucleus. On the other hand, some cells, like muscle cells, can have more than one nucleus. Multiple nuclei mean multiple copies of genes, which are needed for production of the large amounts of protein and enzymes that are needed for muscle contraction. Attached to the nuclear membrane, we find the endoplasmic reticulum. The endoplasmic reticulum is basically a continuous membrane system that is mostly involved in the processing of molecules that are produced by the cell. It is divided into the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes attached to it. Ribosomes are in charge of protein synthesis, so the rough endoplasmic reticulum is involved in this process. It basically provides a place where some ribosomes in the cell can then make proteins, and then it helps with the folding and modifications of these proteins into their final structure. It also then packages the proteins it has processed into packages called vesicles that will then be transported to the Golgi body or Golgi apparatus. Now, the smooth endoplasmic reticulum does not have any ribosomes attached to it, so it is not involved in the manufacture of proteins at all. Instead, it is involved in a variety of other functions, starting with the manufacture or the synthesis of lipids, like, for example, lipid hormones called steroids, as well as the synthesis of some carbohydrates. The smooth endoplasmic reticulum is also involved in the breakdown of drugs and toxins, a process called detoxification. And a third function is one of storage. Some cells in the body will actually use the smooth endoplasmic reticulum to store calcium ions, especially muscle and nerve cells, which use those calcium ions in order to function properly. So getting back then to the idea that form determines function. Liver cells, which are responsible for detoxification of the body, have an impressive amount of smooth endoplasmic reticulum. And cells of the ovaries and testes, which produce the steroid hormones estrogen and testosterone, contain large amounts of smooth endoplasmic reticulum as well. Now, once the endoplasmic reticulum has modified the proteins or lipids, they are then sent to the Golgi body in a vesicle. Now, the Golgi body is sometimes called the Golgi apparatus or sometimes called the Golgi complex. And the Golgi apparatus is basically a series of membranes that are shaped like pancakes. And these single membranes that make up the Golgi body, and basically all the other membranes in the cell, are made up of a phospholipid bilayer, just like the plasma membrane. So the Golgi body basically absorbs vesicles from the rough endoplasmic reticulum, gathers the molecules inside them, and then combines them to make molecules that are more complex or that needed some finishing touches in order to complete them. It then takes those big molecules that it has modified, packages them up again into vesicles, and either stores them for later or sends them to the plasma membrane so that they can be excreted out of the cell. The Golgi body is also the organelle that builds lysosomes. Lysosomes are basically the cell digestion machines, and we will discuss those in a minute. And so once again, getting back to the idea that form determines function. Since the main function of the Golgi body is to package substances that are needed to be secreted out of the cell, cells in body systems that are basically in charge of secretion will need extra Golgi bodies. Like, for example, cells in glands that produce either hormones or enzymes. Like, for example, the pancreas that produces both hormones and enzymes. The pancreas produces two hormones, insulin and glucagon, and a whole bunch of enzymes that are responsible for digestion. Our brain and nerve cells are cells called neurons. And neurons basically produce little proteins called neurotransmitters that are used in order to transmit nerve signals from one neuron to another or from one neuron to a muscle, say, for example. And a third example are special cells called B cells that are part of our immune system. B cells produce little tiny proteins called antibodies. And these antibodies are basically secreted out of the B cells into the blood in order to help fight infections. And in case you didn't know, vesicles, as shown here, are just little membrane packages that mostly transport material throughout the cell, but sometimes also store them. Now, lysosomes are basically specialized vesicles that hold a variety of hydrolytic enzymes. These enzymes are proteins that are first created in the rough endoplasmic reticulum, are then packaged into vesicles, and sent to the Golgi apparatus. The Golgi then does its final work to create the digestive enzymes and then pinches off a lysosome. And from there, the lysosome will float in the cytoplasm until they are needed. Like, for example, to break down invading bacteria or to digest some food, depending on the type of cell that it is. Also, it can be used to recycle old organelles, like, for example, this mitochondria right here. Sometimes the lysosomes are used to break down the cell itself, a process of cell suicide called apoptosis. So, what might trigger apoptosis? Well, there are a few reasons for a cell needing to self destruct. Like, for example, when a body part is no longer needed, like in tadpoles that no longer need their tails, or when the membranes between our fingers need to be removed during development. Another reason for self destruction is what happens when a cell becomes cancerous, or at least starts to become cancerous. That basically triggers a self destruct mechanism in the cell that essentially prevents tumors from forming. Cancer and tumors basically occur most of the time when too little apoptosis occurs. This results in malignant cells that will not die and will accumulate and will then go on to destroy body parts. Now, vacuoles are also membrane packages. However, they do not move cell products around or contain enzymes. They basically serve a very simple storage role. They are there to store water or food or waste or fat. Plant cells tend to have one very large vacuole that is full of water, and that vacuole helps the plant cell maintain an internal pressure. Basically, plants use their cell walls to provide support, but the size of the cell may increase or decrease depending on how much water is present. So plant cells do not actually shrink because of changes in the amount of cytoplasm, but mostly because of changes in the amount of water that is in their vacuoles. When plant vacuoles are empty of water or when they lose a lot of water, the plant may become droopy or wilted. And when the plant gets watered again, the vacuoles will fill up again and the plant can regain its structure. Now, vacuoles can also play a special role in protists. These single-celled organisms live in freshwater environments and because of that are constantly taking up water. So they have these special vacuoles called contractile vacuoles that essentially can contract and relax in order to pump any extra water out of the cell. And what this does is that it ensures that the cell does not end up too full of water, which can actually cause the cell to burst if the cell does not find a way of getting rid of that extra water. And so contractile vacuoles basically are very important in these freshwater protists because it helps them from basically dying by bursting from too much water. And the final part of the endomembrane system is the ribosomes. Ribosomes are the protein builders or the protein synthesizers of the cell. They're like the construction guys who connect one amino acid at a time and build long chain polypeptides. Ribosomes basically come in two parts that assemble when they need to get to work in order to read the genetic code and build a protein. Both parts of a ribosome are made out of a combination of protein and RNA, and they're made in the nucleolus. Ribosomes are special because they are found in both prokaryotes and eukaryotic cells. And because, unlike any other cell structure we've covered so far, they are not surrounded by a membrane. And remember, prokaryotic cells cannot have any membrane-bound organelles, but ribosomes do not have a membrane, so they are actually found within prokaryotic cells. Now, in eukaryotes, ribosomes can be found free-floating in the cytoplasm, but also attached to the endoplasmic reticulum. Next is the mitochondria, the powerhouse of the cell, because the mitochondria's job is to produce energy for the cell in the form of a molecule called ATP. The mitochondria has two membranes, an outer membrane that is smooth and an inner membrane that has many folding called cristae that increase the surface area of the mitochondria. And most of the reactions involved in ATP or energy production are found in that inner membrane. So the more foldings, the more energy the mitochondria can produce for the cell. And it's not just the amount of cristae foldings that can differ between cells based on their energy needs, but also the number of mitochondria found in each cell. Cells that use a lot of energy, like for example our heart muscle, and pretty much all muscle, will have more mitochondria than average. Human heart muscle, for example, will have over 5,000 mitochondria per cell. This is a lot compared to the average of about 500 mitochondria per cell. And liver cells also have a lot more than average, but not as much as muscle cells, with about 2,000 or more mitochondria per liver cell. Now, chloroplasts are another type of organelle that are kind of related or contrasted with mitochondria. And chloroplast is the organelle that is responsible for photosynthesis and is found exclusively in plant or algae and pretty much any other photosynthetic eukaryotic organism. Chloroplasts contain the green pigment chlorophyll, which is used during photosynthesis to make sugars. And chloroplasts, like mitochondria, also have a double membrane. And they also have these little stacks of membranes inside that called thylakoids. And these are the areas that provide that extra surface area that is needed for capturing light into usable energy during photosynthesis. And all of these organelles are not simply floating freely within the cell. Instead, they are anchored down and held in place by a dynamic system of protein filaments that make up the cytoskeleton. The cytoskeleton gives the cell structure. They also help during cell division. For example, spindle fibers are part of the cytoskeleton. They also allow for movement of organelles within the cell, as seen in this animation of a vesicle being moved by a kinesin protein from one part of the cell to another. And it's basically moving along the protein fibers of the cytoskeleton. The cytoskeleton is also involved with cell movement. Whenever a cell needs to move, it's the cytoskeleton that's usually responsible for that. Like, for example, in the case of structures called cilia and flagella, which we'll discuss in a little bit more detail in a second. Microtubules are what centrioles are made of. They are found only in animal cells and produce spindle fibers that move chromosomes around during cell division. And both cilia and flagella are also made of microtubules. Flagella are long, thick tails that whip around and sometimes twirl, pushing a cell forward. And cilia are little hairs that you might see on the outside of a paramecium or other protists, and they flap back and forth to help the cell move. Protein fibers are also found inside structures called pseudopods. Pseudopods are not permanent structures of a cell. They are temporary arm-like projections of a eukaryotic cell membrane. They are filled with cytoplasm, and pseudopods or pseudopodia primarily consist of actin microfilaments. They also may contain microtubules or intermediate filaments, but they're believed to be mostly made out of microfilaments. And the word pseudopod basically means false feet. It comes from the Greek words pseudos and podos, meaning false and feet respectively. And as such, they are often used for movement by some cells, and also for feeding or for taking in molecules from the outside of the cell. This amoeba, for example, is using pseudopods for moving around, but in this particular video, or very short video clip, it is using its pseudopods to feed on this paramecium. Now, the cytoplasm is the fluid that fills a cell and is found between the cell membrane and the nuclear membrane. The fluid in the nucleus is called the nucleoplasm and is separate from the cytoplasm. Each of those fluids have very different composition. The cell organelles are suspended in the liquid portion of the cytoplasm called the cytosol. And as we learned a few minutes ago, these organelles are held in place and moved around by the cytoskeleton. The cell wall is a structure made of sugars that surrounds the cell membrane. In plants, it is made out of cellulose, while in fungi, it is made out of chitin. And while the cell wall protects the cells and gives them shape, they also allow a plant to grow to great heights. And finally, let's talk about the cell membrane, also known as the plasma membrane. The plasma membrane is the boundary that separates the inside of a living cell, also called the intracellular space, from its surrounding or extracellular environment. And the main job of the plasma membrane is to control what can go in and out of the cell. Because it allows some substances to enter the cell but not others, it is described as a selectively permeable membrane. Cell membranes are made primarily of a double layer of phospholipids, with some other molecules either embedded in between the phospholipids or in their surface. Molecules like cholesterol or proteins, for example. So let's look at the different components of the plasma membrane one at a time. And let's start with the phospholipid bilayer. So recall from our lesson on lipids that phospholipids have at one end a polar head that is hydrophilic or water-loving. That means that it interacts well with water. At the other end, they have two non-polar fatty acid tails that are hydrophobic or water-hating and do not interact well with water. Because of these unique properties, when placed in a liquid environment, phospholipids then tend to arrange themselves into something called a phospholipid bilayer, so that only the hydrophilic heads interact with a watery environment and the hydrophobic tails crowd inward away from the water. In 3 dimensions, it would look a little more like this, with the heads facing the watery exterior and interior of the cell, and the hydrophobic fatty acid tails sandwiched in between the phosphate heads away from the water. Another thing that you need to know is that the membranes that surround the organelles inside the cell are also made of a phospholipid bilayer. For example, let's take a look at this vesicle. This vesicle comes from the Golgi body, and it is carrying a cell product that was produced at the endoplasmic reticulum, packaged into a vesicle there, and then sent to the Golgi body, where it was finished and repackaged into another vesicle. Now it is headed towards the plasma membrane, where it will deliver its contents to the outside of the cell by fusing with the plasma membrane. Since the membrane of the vesicle is made up of the same phospholipids that makes up the plasma membrane, it simply becomes part of the plasma membrane itself. Also, contents from outside the cell can be brought into the cell by forming vesicles from the plasma membrane. There are a number of different proteins embedded in the membrane or on its surface, and they serve a variety of functions. Some are involved in the transport of substances across the membrane. Some receive chemical signals from outside the cell and then translate the chemical signals into some type of intracellular action. Also, enzymes in the membrane can do the same thing they do in the cytoplasm of a cell. They can transform a molecule into another form. And anchor proteins can physically link intracellular structures like, say, the cytoskeleton within the cell with the membrane itself, or link the membrane with structures outside the cell. Some of the proteins have sugar chains attached to them, forming glycoproteins. Glycoproteins have a variety of functions, but mainly serve as recognition signals to other cells. So the carbohydrate or sugar chains that are attached to these proteins allow for some unique shapes, which then can act as markers that can be used for cell-to-cell recognition. Like, for example, it allows cells to distinguish self from non-self, which is the basis of our immune system. The sugar chains also serve a role in cell-to-cell adhesion, allowing cells to attach to other cells to form tissues. And while glycoproteins are proteins that are covalently bonded to carbohydrates, glycolipids then are lipids that are bonded to carbohydrate molecules. And cell communication and adhesion are not the only roles of glycoproteins and glycolipids. For example, glycolipids are used for electrical insulation in nerve cells. They are found in high numbers in an insulating sheath called a myelin sheath that is found surrounding a part of the nerve cell or neuron. And phospholipids are not the only lipid molecule found on the plasma membrane. Embedded throughout the plasma membrane is a lipid molecule called cholesterol. Cholesterol lends support to the membrane and helps regulate its fluidity. And speaking of fluidity, the current view of the plasma membrane is called the fluid mosaic model, which describes the membrane as both a fluid and a mosaic. But what do those words mean? So let's start with the word fluid. The cell membrane is fluid because the phospholipids and the proteins that are embedded between them are free to move. The components of the plasma membrane do not stay in one place. They are in constant motion. So this fluidity is due to the structure of the phospholipids and is also helped along by the presence of cholesterol molecules. And that is because the phospholipids of the plasma membrane are not static. They don't stay in place. They're in constant movement. They move rapidly and constantly past each other in a form of lateral movement, and also very occasionally they flip-flop with their bilayer partners. So a factor that leads to this fluidity is the nature of the phospholipids themselves. The ratio of saturated and unsaturated fatty acids within the phospholipids helps determine the fluidity of the membrane, especially at cold temperatures. In their saturated form, the fatty acids in the phospholipid tails are relatively straight. In contrast, unsaturated fatty acids contain some double bonds, which then bends the tails. So when it is cold, the saturated fatty acids with their straight tails are compressed by decreasing temperatures. They press in on each other, making a dense and fairly rigid membrane. But when unsaturated fatty acids are compressed, the kinks in their tails sort of push or elbow adjacent phospholipid molecules away from each other, maintaining some space between the phospholipid molecules. And this elbow room helps to maintain the fluidity in the membrane at lower temperatures. So the composition of the phospholipid bilayer is particularly important in a cold environment. A cold environment tends to compress membranes composed largely of saturated fatty acids, making them less fluid and more susceptible to rupturing. Many organisms, like fish for example, can adapt to cold temperatures by changing the proportion of unsaturated fatty acids in their membranes in response to the lowering of the temperature. In other words, when it gets cold, some organisms increase the amount of unsaturated fatty acids in their membranes in order to keep their membranes fluid. In animal cells, another factor that keeps the membrane fluid is cholesterol. Molecules of cholesterol can be found embedded within the phospholipid bilayer in the membrane. The role of cholesterol is to basically dampen the effect of temperature changes in the membrane. So cholesterol acts a bit like a buffer, preventing lower temperatures from lowering the fluidity of the membrane, but also preventing higher temperatures from increasing the fluidity too much. What cholesterol does is it extends the range of temperatures in which a membrane can remain fluid and therefore functional. So at lower temperatures, cholesterol increases the space between phospholipids, which then keeps the membrane fluid. While at higher temperatures, cholesterol restrains the movement of phospholipids, reducing the fluidity of the membrane, essentially keeping it from melting. One of the reasons that the plasma membrane is fluid is actually due to its mosaic nature. The plasma membrane is a mosaic because it has many different types of molecules embedded throughout. And this mosaic characteristic of the membrane actually helps the plasma membrane remain fluid. The integral proteins, those that are embedded in the bilayer, exist in the membrane as separate but loosely attached molecules that are able to help keep the membrane fluid. And so the fluid mosaic model helps explain how the plasma membrane is both a fluid, with components that are in constant motion, and a mosaic with many different types of molecules embedded throughout. So that's it for now, and I'll talk to you soon.